Hi students. In previous session under molecular basis of inheritance, I introduced the most interesting molecule without which our survival, our existence is zero. And that molecule is nothing but deoxyribonucleic acid, abbreviated as DNA. This DNA is chemically a polynucleotide having a polymer of nucleotides. So monomer of DNA is nucleotide. Each nucleotide is composed of nucleoside and phosphoric acid. Phosphoric acid is H3PO4. It is a phosphate donor. Nucleoside is composed of a pentose sugar and a nitrogen base. Deoxyribose sugar having 5 carbons, 10 hydrogens, where 1 oxygen is less here. So its molecular formula is C5H10O4. This deoxyribose sugar develop N-glycosidic linkage with nitrogen base. This nitrogen base is either purine or pyrimidine. The purines are heterocyclic molecules with double ring structure. They are adenine and guanine. Adenine represented by capital A, guanine represented by capital G. Pyrimidines present in DNA are cytosine as C, thymine as T. In this session, I'll just explain you how the nucleotides are formed and how the polynucleotide is built up, so that it is easy to understand double helix structure of DNA, which is a compulsory structure expected for 5 marks in board exam. As I said, the sugar is deoxyribose. It is a pentose sugar. This is the structure of sugar moiety with 5 carbon atoms. Here, the 5th carbon with functional group can establish linkage. Okay, this linkage is with phosphoric acid. So oxygen of phosphoric acid develop linkage here. So this is called ester bond. This sugar, I'll mention here S, this is sugar, this is carbon 1, carbon 2, carbon 3, carbon 4 and carbon 5. This represents 5' end of a polymer when it polymerizes. A nitrogen base is linked. So the bond present here, this is glycosidic bond. And I'll take nitrogen base as purine. And in purine, I have taken this adenine. Sugar, phosphate group, nitrogen base. This represents a nucleotide. Now, when the adjacent nucleotides, okay, let us consider now the next sugar polymerization initiated. This is pentose sugar. This is 5th carbon H, and here this will be a phosphate, okay, showing ester bond. So there are 2 ester bonds to link two sugar molecules. So it is known as diester linkage. So this forms a sugar, and other side I'll write a nitrogen base, and this is a guanine. So this linkage is glycosidic bond. Like this, the polymer start developing diester bonds here. When I draw this structure, okay, let us consider this is a line representing sugars. They are interconnected by phosphodiester bonds. Let me complete. At least 4 nucleotides we can take in a chain, so we can include 4 nitrogen bases there. I already told you, line represents sugar backbone, outside phosphates, and now inside I will write here adenine. This is guanine, this is cytosine, this is thymine. Clear? This is 5' end, this is 3' end. This is one polynucleotide, one strand of DNA. DNA is always double-stranded, where another chain, this is sugar backbone. Now here phosphodiester linkage. When a bond develops between C and O, it is ester bond. So here also I have taken a polynucleotide. Now here the arrangement of sugar, I'll just explain here. The sugar is inverted like this. Okay, this is carbon 1, 2, 3, 4, and this is the 5th carbon. So this end 3', and this end we call 5'. Here this is glycosidic linkage, and here ester linkage. So here sugars are inverted. So when the two strands face each other, due to its anti-parallel arrangement, we can see here in front of purine there is a pyrimidine, and in front of pyrimidine there is a purine. Again there is a specification here. Adenine always have pairing with thymine. Guanine always have pairing with cytosine. Cytosine always have pairing with guanine, and thymine with adenine. Now the third linkage develops. There are 2 hydrogen bonds here. Here 3 hydrogen bonds. Here 3 hydrogen bonds, and here 2 hydrogen bonds. Clear? Now this we call 3' end, and this we call 5' end. So, chemically, DNA is having 3 chemical compounds. Deoxyribose sugar, it is nothing but pentose with 1 oxygen less. It will develop glycosidic linkage with any one nitrogen base, where bases are 4. So variation lies in the base, where sugar and phosphate remain same. So variability in the particular DNA structure is because of the pairing. Pairing is between nitrogen base, and this pairing, you should not forget that adenine pairs with thymine and guanine pairs with cytosine, and vice versa. Total 3 bonds I told. The question expected for 3 marks. Name the chemical bondings found in DNA molecule. First, you have to say about N-glycosidic bond. Just recall where it develops. Second, phosphoester, or you can call phosphodiester bond. This will help in polymerization. And third, hydrogen bond. This will help in formation of base pairs. The weakest bond in DNA, this is neat point. The weak bonds in this DNA are hydrogen bonds. The strong bonds are ester bonds. Slightly less stronger are glycosidic bonds. So when this DNA molecule exposed to high temperature, denaturation is resulted. During denaturation, the weak bonds will dissolve, and that's why the two strands of DNA separates. So denaturation of DNA may result into mutations. You already heard what mutation is. And wrong mutations definitely causes genetic disorders in the organism. Hope it is clear what exactly DNA and its chemical composition. So before we begin, the physical structure of DNA. Okay, in tomorrow's session, you please remember the arrangement of nucleotides, how the polynucleotide is arranged, and once it develops hydrogen bonding, okay, it looks like a ladder. This is how the DNA forms: 5 prime, 3 prime, 3 prime, 5 prime. You can call 5 dash 3 dash or 5 prime 3 prime. This is anti-parallel arrangement of two strands of DNA. These are hydrogen bondings. This is just diagrammatic representation of DNA. It looks like a ladder now, and this ladder is helically coiled. Clear? When I take now the two pens together, okay, with 2 finger distance, this is the idea to sketch the double helical structure of DNA, right? You have to keep the 2 points. 2 fingers should be in the center, right? And then you have to draw the structure like this. Okay, you need practice to draw this structure, but if you draw like this, there's a perfect structure of DNA having double helix, helically twisted. Okay, now here the green colored and orange colored. Green colored is sensible strand, orange colored is antisense strand. Due to coiling, okay, they forms the helix. So this we call one helix. The length is 34 angstrom. This is the diameter, 20 angstrom. In one helix there are 2 loops. In each loop, okay, there are 5 base pairs. 1, 2, 3, 4, 5. 1, 2, 3, 4 and 5. Okay, so in one helix, how many base pairs are there? 10 base pairs. Don't get confused. Base pairing is between purine and pyrimidine, and they are forming hydrogen bonds. This is called minor groove, and this is called major groove. This is the representation of Watson and Crick's double helix model to learn the structure of DNA. For this, They awarded with Nobel Prize in 1953, and this is based on the photograph which is collected and taken by Wilkins and Franklin using X-ray crystallography method. In tomorrow's session, I'll explain the typical features of this double-stranded helically twisted DNA molecule. Thank you very much.